So let me just tell you a little bit about myself. So I'm Dr. Hannah Saw. I'm the CEO and one of the co-founders of PharmEnable Therapeutics. I've got about 25 years in the industry, working originally as a medicinal chemist at Millennium Pharmaceuticals and then at Aztec. So I was lucky enough 20 odd years ago to work on that PKB inhibitor that now with the ICR and AstraZeneca has just got approval, and really nice celebration down in London at St Paul's just a couple of weeks ago. So that was really nice. I think as a scientist, that's what we're all here for, to make an impact for patients. After I really like business and science, so I went to work down in London. I went to the dark side and worked as a management consultant, I would say, which was really great. I got to, see what was happening across healthcare and drug discovery, rather than just very narrow into one particular area. And there I could see, I used to do talks on lots of different modalities. Fantastic, there's talks here obviously today about, biologics, cell and gene therapy. But from my background, the little old small molecule, everyone was like, that's yesterday. So that really got me motivated to think, well, what can we do to make small molecules sexy again? So I'm really here I'm here for the small molecules. So really, what can we do to push the boundaries of a small molecule? I have a little prop, so this is for the non-chemists here, just so you can see. This, I don't know if we've got molecular model here. I like to bring a molecular model. This is paracetamol, very old, obviously, first made in 1878, I think it was.
Show the camera, Hannah.
Okay, this is paracetamol. Pretty, amazing in its time. I don't know if you can notice one thing. It's a molecular weight of 151. But I don't know if you can particularly notice one thing. It's pretty flat. So really, really flat molecule. And I think this is where I'm really excited about. We're all here. We're all three-dimensional. We believe that small molecule drugs actually need to be as well. And as a chemist, making things 3D is really difficult. Andy is nodding in the background there. It is really challenging to make three-dimensional small molecules, and that's what we're all about. So as I said, we're really, really pushing the boundaries, reimagining what can you actually achieve with a small molecule therapeutic. We're doing that through AI. So we're really looking at accessing the inaccessible, and I'll explain a little bit more about what that actually is. So as I said, we're, when we say targeting the accessing the inaccessible, we're really looking at trying to replicate the specificity of a biologic, but in a really powerful small molecule form. And in particular, going after the targets that are inaccessible to biologics. So in particular, things in the brain, so passing the blood-brain barrier, and also inside the cell. So we have a really amazing AI-enabled drug discovery platform that really enables us to unlock these new areas of chemical space. We call it our 3D accessible chemical space. We have a rapidly growing pipeline. So we have in the neurology and oncology disease areas. We're a spin out from the Cambridge University. We have an amazing supporters and investors that are here, obviously at O2H as well. And so to date, we've raised just over $10 million. And later this year, we'll be going out for our Series A. And that will be to advance our programs towards the clinic. So I think everyone can appreciate antibodies, really amazing drug modality, really have been very transformational, some of the largest, obviously, selling drugs, which is brilliant. But I would say that many targets are still inaccessible. Obviously, as I said, the CNS and the blood-brain barrier is a real challenge for antibodies and intracellular targets as well. So these are areas where we're really excited to focus in on. And so the way how we see the world is, there's a lot of fantastic new modalities. I was out at JPM, and ADCs, amazing, really great, exciting area, and bispecifics. And then you've got the new world, PROTACs. We're also in the PROTAC space. Really excited on what we can do with PROTACs. Cell-penetrating peptides. But we really believe, we're really excited about the AI world and what we can actually achieve there. And really looking at small molecule therapeutics that have got antibody-like specificity. And that's not trivial. That is incredibly difficult and challenging to do. So very quick introduction to the platform, which is ChemSeq. And what it is, so chemical space is the most vast concept you can possibly think of. It's more than the grains of sand. So you can't actually physically have enough atoms to make all the possible combinations. It's 10 to the power of 62, so it's huge. So what we've developed is a mathematical model of what actually even is chemical space, so that we have this understanding, so that when we're trying to design a new molecule for a given drug target or polypharmacology, multiple drug targets, we know exactly where to actually search in chemical space. We're then able to populate that with novel three-dimensional small molecules. And this is the thing. So it's the coded knowledge that we've been working on that enables us to build these three-dimensional small molecules. And as a chemist, using generative AI, you can design anything. You need to be able to make it. You need to be able to advance it and take it through. And so, once it actually gets to being a drug, the CMC needs to be something that is not prohibitive. And then it's really about, when we've created this vast chemical space, it can be billions of molecules at any particular target. They then need to filter that down really efficiently. And so it's drug discovery, if anyone's done it, it's like whack-a-mole. So generally, as soon as you've got one property, another one pops up. And I think this is a key tool that we use in order to get over that. So I'm just going to tell you very quickly about our lead program. This was just the hit discovery part and how quickly we were able to do it. So I'm all about efficiency. So this is a rationally designed dual kinase inhibitor. There are known single inhibitors on there, but that has seen pathway redundancy. So the key thing here is a dual inhibitor, and to date, that has been eluded everyone. So people have been able to get pan kinase, but not this very specific dual inhibitor activity. And I just wanted to highlight 36 in our hit discovery molecules. We're low 10 nanomolar. We've got multiple validated theories within that set, completely unknown scaffolds, and then we've got the selectivity profile we want. So we've got that amazing properties, which in a really short space of time is pretty impressive. Yeah, thank you. And so this program is now in hit to lead, and we're going into lead optimization later this year. We've got a brilliant team and yes, built up. So we're actually a remote first company. So we believe in getting the best talent no matter where they are in the world. And so, we have people across the UK and also across Europe. So the leadership team here, so I worked with Kyla at Millennium and at Aztecs actually. So it's a very small world in terms of the thing. And then we've got a fantastic board. The most recent addition was Ian as well. Again, actually, he was involved with the PKB program. So there's a lot of sort of common themes there. And then very strong scientific advisory board covering all different aspects of drug discovery. So I don't want to leave you, but one of the things that we really believe is the only way really to discover the limits of what's possible is definitely to go beyond them into the unknown. So we're really excited that we will be starting to raise our Series A financing later this year, the second half of this year. to take our programs. I haven't said about the different programs that we've got on. There's a lot of work in ongoing at the minute with some really exciting deals, and we have some other co-discoveries with, for example, Actaris as well. But yeah, I don't want to hold you up from your cocktails, but yeah, thanks very much.
Thank you.